to see me in my office hours at any time, so I will hold them online as well as in this classroom here, except for I won't be in the classroom at night, so then you'd have to go and see me online if you're on Iowa time or if you're on GM time or some other time. But if you do have questions, feel free to check them out. And I am also available via Google+, Skype, any other method you like. Just let me know. You might need to email me for my Skype address if you do want to meet via Skype. Any questions before we start today's lecture? Okay. So I also wanted to mention that on the schedule website, there are links to all the papers that we are discussing, as well as some additional readings. So this is the paper that we discussed on Monday, and we will spend most of the time today finishing up that paper. We will briefly introduce this paper, and then on Friday, we will meet if you're on UI, the third floor computer lab in McLean Hall. So that's 301 MLH if you're at UI. If not, you can still join online. We'll still have the same standard classroom. If you have access to MATLAB, you can follow along and run with it. We will demonstrate two programs. One is the MATLAB version. The nice thing about the MATLAB version is it comes with MATLAB, so that means you can use any additional functions. So for example, we mentioned one of the choices you get to make is how to cluster the data. There's lots of different softwares that are compatible with MATLAB that will cluster data, so that gives you more options. The bad thing about it is it's associated with MATLAB, which of course has its own issues. There is also a web tool, but then you have to use, the choices embedded within that web tool, although you can also output some things too and then use it to in other type of stuff. So we'll demonstrate both of those. We are only spending 1 week on the Mapper software, so after this you can play around with the Mapper software, and if you have additional questions and we want to discuss it again, just let me know and I'd be happy to go into more detail. But I figured this is enough so that you can start playing around with it. The other software that we will be taking a look at a variety of other softwares involving persistent homology that do a lot more than just the mapper, so they're sort of disjoint in many ways. But there's a lot of documentation on the other softwares that we'll take a look at. But let me know if you do want to discuss more of the mapper software, because as you'll see today, we get some very interesting results from it. Okay. So again, this is the paper that we are discussing. So just to quickly review what we did on Monday. We started off with a data set. In our first example, it was a point cloud. We'll look at 3 other examples, but basically, as long as we have a bunch of data, we're in good shape. It helps if we understand the data, because we then will choose a filter function. So this is our filter function. So in this particular case, we just projected onto the x-coordinate. So took a point here and projected onto the x-coordinate. But we can go into any codomain that we want. In the 3 examples, the real ones we'll be looking at today, we'll be using 2 filter functions. So when we combine them, they go into R2, but there's a lot of different choices one can make. So you have to choose a filter function. Usually, it's not just projecting onto a coordinate. Normally speaking, you want to understand your data, and if there's a property that you want to analyze, that's a good choice. Otherwise, principal component analysis, anything where you're projecting nicely, where you're getting lots of information, is a better choice than arbitrarily choosing the x-coordinate. But for this example, it illustrates the stuff nicely. So once we have our filter function, we can then take our codomain. So we take our codomain R and we start covering our codomain. So I can cover it with a bunch of overlapping intervals. So we'll cover it with a bunch of overlapping intervals, and then we project back. I didn't do a very good job in aligning, so if I had moved this thing over, that we project back and divide up. So we now have divided up our data into overlapping bins. So we have these overlapping bins, and now that we've got these overlapping bins, we can then cluster each bin. So for this particular bin here, we see that there are 3 things. That becomes 3 vertices. In 3 dimensions, we've got this bin here, and the thumb is actually separated out in 3 dimensions. And so we have 2 bins here. So we cluster, and that gives us our vertices. And then we will join 2 vertices, we'll join 2 clusters if there is a non-empty intersection. So between these 2, they did intersect. So there's some intersection between these 2 clusters, so we join them up. And so that gives us our nice visual representation of our hand or of our data. So that's our basic mapper software. So when we do the mapper, we'll definitely be choosing the filter function as well as the bins as well as the clustering. One can generalize the B and C, and as long as we have data and we can get it into any type of overlapping bin. So as long as you can group your data into these overlapping bins, you can still do the same thing of creating a vertex. You can still cluster each bin, create that vertex for each cluster in each bin, and then create edges joining the clusters. So remember, we did, again, make many, many choices. So that does allow us to explore data from a variety of different mathematical perspectives, as well as a lot of different applications. So depending upon your application, if there's properties you want to focus on, you get to choose the properties, and you can see how the various different properties are affected by, how the graph is affected by your different choices. Of course, when we do have lots of choices, we can, get a lot of different results that may or may not actually be applicable. They may not be real. We might have some false positives. But if they persist over a variety of choices, then there's a good chance we do have, that the result is actually valid. So just again, to reiterate our choices, we first have to have a data set to enter in. I'll provide some data sets on Friday, or you can use your own. The data sets generally have to be cleaned up, so there's been a lot of work just to create the data set and clean up the data set. The web version, for example, will not allow missing values. If you've got any missing data, you have to fill in those values with something. Then we can choose a variety of different functions. We'll take a look at that to project it down. Then we have our choosing our overlapping bins. Generally speaking, we will take a look at one or more filter functions that go into R, and then we will partition them with things of equal length. And so we'll usually just choose the length of an interval, and they'll all be the same length, as well as the percent overlap. So we have 50% overlap between the intervals, 80% overlap, things like that. Then we have to choose a way to cluster, and after that choice is made, then we know exactly how to create our graph using the vertices and edges. Any questions so far? And just to reiterate, we did have some nice things about topological data analysis that makes it superior to any other data analysis out there. No, one should look at these as a tool, but there's a lot of tools, and comparing them to the benefits of this tool versus the benefits of the other tools is a nice thing to do. But it does give you a new tool, and one of the nice things about that is it is coordinate free, so we will be able to compare data derived from a variety of different platforms, which will be very nice. We'll look at that in particular when we take a look at the breast cancer data. I have a quote from one of the papers that says, we compare it to this other stuff, but comparison, we can't really do it because of all these differences between platforms. That you'll notice we'll actually be able to do that with the topological data analysis. So that makes it very nice. It's also invariant under small deformations. If my point is here and I move it over here, if my point is here and I move it over here, it'll still probably end up in the same cluster. And even if it doesn't, it still might not change where we've got the edges. And so there's a good chance that small perturbations won't affect your output. And then the other thing is we do get a compressed representation of the shape. So we do get a graph, a network that we can actually see. There are other types of data analysis besides topological data analysis that will give you graphs, but we definitely do get graphs in this thing, which gives you a little bit. It's easier to see the relationship between all the data points that get clustered into here versus all the data points that get clustered into here. I know over here they're far away. So it does give me some sort of relationship that is easier to see when we have this graph that we can see. Okay, so that I believe reviews from last time, and we can now talk about applications. We're going to start off with going backwards. Basically, I want to save the best application for last, and so we'll do the simpler applications first and talk about basketball. So in basketball, you can actually get this data off the web, and you can get the rates per minute played. So different basketball players might some might play for 20 minutes, some might play for 40 minutes during the game. And so you've got rates per minute played of rebounds, assists, turnovers, etc. There are 7 different, so we've got 7 different properties that were downloaded to be analyzed for 452 players. So that means we have 452 points, so 1 data point for each player. And the data point is a vector in R to the 7, so this is our real numbers right here, where our first coordinate is rebounds per minute, our second coordinate is assists per minute, etc. So we get this vector in R to the 7. We then when we want to analyze the data, we will use the variance normalized Euclidean distance. So if, for example, we take a look at, we could just calculate distance and take the norm for that for our distance. But that could distort things a little bit, and the reason it distorts is in terms of points scored versus fouls made. One hopes that the player is scoring more points per minute than they are doing fouls per minute. And so if you're trying to analyze the data, if the points scored, they probably vary a lot. Kobe Bryant is probably doing a lot of scoring, so he's probably up the top in terms of the scoring, versus, some people that's not their job to score, so they'll have quite, that's not their main focus, so they'll have far fewer baskets made. Versus personal fouls, we hope that that's a pretty slim range. People aren't, doing many fouls. We hope, and so that would be a smaller range. And so often in data analysis, whether you're talking about basketball or anything else, you frequently will normalize the data so that the variance, here it varies quite a lot. Here's a small variance, so that all the variance is the same. And so that way, this doesn't dominate your results in terms of your distance. And so you can see both the fouls as well as the points scored. Our clustering is single linkage. This is a standard clustering mechanism, and it's very similar to what we talked about in terms of I have my different points here, and points that are close together, I will group them together. Points that are far away, I don't. And so we talked about, growing balls to find out what's the best sort of grouping. So it's very similar to actually just creating the simplicial complex where you're just getting the edges. And there's a lot of different techniques So standard things to do to create this single linkage so that you cluster things that are close together and not things that are, far apart. So that's the basketball data. And so the filter will be the principal and secondary single value decomposition. So single value decomposition is often used in data analysis. And that's basically because, in our hand example, we could see the hand, so we could see that projecting along the x-coordinate was the best thing to do. When you have high-dimensional data, you don't know where is the best direction to project, but we can see that if I take a look at this data right here, if I take a look at the line through here, that's where it varies the most. And so with your single value decomposition, it's basically telling you where to best project to get the most information. And so with the principal, you do your first projection, then you do a second projection, which will be 90 degrees orthogonal to the first projection, where you get the second variance. With this two-dimensional data, it's obvious I just take the one that's perpendicular, but I could have, my third value would have been the one coming out of the plane where I have no variance whatsoever, so I would have no information. I don't want to project going out of the plane because all my data is within the plane. So that's basically what is done in this example and in other examples that we're looking at, is we are again projecting the data, but the choice of where to project is used using singular value decomposition. So we do take our data and we project onto our principal and our secondary. And since we are dividing up, the principal one, we divide up the secondary one, then we take each of these things here, project back into our data. We will get overlapping sets, which we, first off, we project back. So let me first project this one here, we project here, that we will then cluster this here, and then if we do have some overlap, we'll then go ahead and join edges, creating our two things. So that is what they did. Let's actually see a prettier picture of that. This is the result. One of the things in terms of the filter function is we can do different resolutions. So we can do a low resolution as well as a high resolution. And by low resolution is we did have, we were projecting onto R2 because we were using two filters. But let me draw this for just R1. The question is, how many intervals do we want to use? So in one case, they used 20 intervals. So we covered our data set with 20 intervals. In the other case, we can get a higher resolution, so the more intervals we use, the more clusters we will see, and so we get a higher resolution if we use more intervals. So that's one thing. You can analyze your data at multi-scales, so we can do multi-scales here, choose just a small number of intervals to do low resolution, choose more intervals to do high resolution, and you can see it does affect our data quite a bit. When we do take a look at this data, one thing to keep in mind is there is software that will draw this out for you, but the software, it's hard to see. So if you've got really good eyesight, we've got this cluster right here and this cluster right here, and you can all see there's an edge between them, can't you? If I look really closely, I can actually see that there is an edge between those two clusters. And so there are actually a lot of edges around here that do join up these different clusters. So we can see that this is one connected component right here. So we've got one connected component right there. And we have several other connected components. It is true that we put the All-Star NBA next to the All-Star NBA second team. So there's likely a relationship, but that's not what was found by just the clustering. It was just drawn there, not randomly, but don't infer too much as to the placement of these different clusters. So for example, if I take a look at the role players they're up over here versus they're down over here. So if I try to compare the graph on the left versus the graph on the right, the comparison is a little bit interesting. There is also some additional information in here. Remember, we use seven different coordinates, so there are 7 different properties that were looked at. But often you will color your graph, and so they color the graph using the points per game. And so the high scorers, we can see, are all colored red, and the low scorers are colored blue. And so you can see that there is a difference in terms of the placement, because the high scorers, the reddish area is over here, the low scorers, the bluish area is over here, versus in this graph, I've got, red here, red here, red here, red here. So the placement is different. So when you're trying to compare graphs, there can be some challenges in terms of visually comparing these graphs. But there is software that will compare graphs for you. So there's, lots of comparisons. That is an interesting research property in and of itself, comparing things. But there are things where you can identify players that might be in here versus in here. And so you can do things to say, oh, this goes, this and this are related because this player is in this cluster and this player is over in this cluster. So there are ways of comparing them, but do be careful about just comparing them by eyesight, because I like graphs, but sometimes it's easy to infer a little bit more into it. We can see that that there is a big difference between our low interval and our high interval, our low resolution and our high resolution. And one of the nice things about our high resolution and one of the nice things about the mapper stuff is you do get these flares. And so you can see that there is a distribution of properties that come out here. That's, I think, always in terms of biology, because my main application is towards biology. So you can imagine in terms of, breast cancer data that we will look at later. There's going to be a variety of distributions, and sometimes being at the end is not, sometimes it's better to be in the middle of this distribution rather than at the ends. And so being able to see the distribution can also help. So it isn't just the groupings that we're interested in, it's also the visualization and these flares that we see that can also give us some additional information. The groupings, though, also give us good information. It has been statistically proven that we're not very good at evaluating people in a completely unbiased way. So when you remove lots of information, so there was recently an article about evaluating piano competition, and they removed the visual part, and the results were quite different than when you actually had the visual. When they removed the sound, the results were actually not as different. They were very similar. So people were actually basing how well people played the piano based upon what they looked like rather than how they sounded. There's been all sorts of data like this. And so having these groups being done by a computer, yes, we get to set a lot of parameters, so we made a lot of choices, but still, as long as we know what those choices are, we understand to some degree these choices, we can then go ahead and say that these groups hopefully have some meaning, take a look at these groups. So for example, let's take a look at the All-Star NBA. I am sure in terms of the All-Star NBA, you've all heard of LeBron James, Kobe Bryant, and Brooke Lopez. Well, I just checked this morning with someone who says that they really are into basketball, and they've certainly heard of these names. I hadn't heard of Brooke Lopez. Well, that's because they're an up-and-comer. So you can, identify up-and-comers. So if you, want to make any bets without money, of course, because, we don't want to do any gambling, you can, Brooke Lopez mathematically. Don't trust me on any basketball recommendations, but it did identify some up-and-comers that I wasn't aware of and other people might not have been aware of that is grouped in terms of this All-Star NBA. You can also note that there are people who are, very disjoint, whose roles are very different than other people's roles, and, that are clustered very far away. And in this case, the clustering is probably since it's, very red, it's very much related to the points per game. So that's our basketball example. Any questions about the basketball example?
Are the labels that are assigned to each group or each cluster, are those derived from like human post-processing, like a human would look at it, or is it part of each player has additional descriptors that are looked at in this TDA?
Excellent question. So the idea is, if we have these sorts of things, these are actually was known by humans. Some of these things were standard roles that were previously identified. Something I forgot to mention, so these are human identified names. The clusters themselves, so we know that the cluster exists, but in terms of what the role is, they can look at the properties. So they look at the properties, for example, it's red, so scoring big is the human name give to it based upon the properties. One could try to automate this a little bit as well, but I believe in this case it was human done and not automated in terms of giving these things. But if you are familiar with basketball, I believe that there are more groups here than what is normally done. So this is also something else that could be useful. So if I ever teach this at an undergraduate level, or if there's people who are interested in this basketball data, we can contact the coaches here and see if they would find these finer subgroups. So this is more refined than I believe what is currently used. And so maybe these finer subgroups would also be of interest. So the human analysis does play a role, and then, how can one use this to maybe better advise the players? So thank you for that question. I had forgotten to mention that we are breaking, these things up into different roles. Based upon, you can see, we don't see the vectors, but one can then analyze the different flares to say this different flare should have different properties. Now let's take a look at the properties of all the people in these various different clusters in this arm here and see what defines them. And to some degree, some of these things are already known, and so we can use what's already known in terms of the players that are in there to help us. And so some of this could be automated because there are people who, we know LeBron James and Kobe Bryant are in the All-Star NBA, and so that is what can be used to identify this group, and thus other people now get identified in that group. Okay. Other questions? Okay. So our second application, again going backwards, is looking at the U.S. House of Representatives voting records. So in this case, this is only the records from when the representative is actually there. So if they're not there, that would be missing data. So we would have, and they could vote yes on something. I would be given a +1 value. They might be there, but they might abstain, so that'll be given a 0 value. Or they might vote no, i.e. nay, which will be given a -1 value. So if on Bill 1 they vote aye, Bill 2 abstain, Bill 3 nay, Bill 4 we have a lot of naysayers, so I'll just put a lot of nays. So we might have a lot of -1s, maybe an abstention, maybe we'll actually get a yes vote sometimes. You never know, it could happen. And so this is what the vectors will look like, is a bunch of 0s, 1s, and -1s. There's a lot of choices we can do. Correlation is one of the main ones. So Pearson correlation is a good one. So that'll be the distance between these things. And our filters will, again, be the principal and secondary single value decomposition. And we will, again, use single linkage clustering. So if we take a look at the results, so these are the results for 2009, 10, and 11. And you can see that in 2009, it appears that there was something going on in 2009 that really unified many of the Democrats and unified many of the Republicans. So the Republicans are red, the Democrats are blue, so that's the coloring here. Versus there wasn't as much things that unified in 2010 and 2011. So then people broke up into many smaller groups. So you can observe the behavior over time, whether or not the individuals change, it doesn't matter. So if this is, we can look at the data from 1887, that's fine as well. You're just comparing what's happening over time. And you can see the number of sub-network parties formed in the various different years over the time. So you can see in 2009 there were very few parties, red Republican, blue Democrat, and yellow being the combined of the two. If we take a look, we're going to take a closer look at this group right here. So I think 2009 was that healthcare type vote, so that sort of explains this division right here. You can analyze the data. This is done automatically to find out what, we have two main groups here. What is it that created these two main groups? So if we take a look at these two main groups, well, we can get to figure out what it is. These were actually the main bills that divided the two main groups. Some of this might make sense. Some of it might just be, based upon who knows what. Oh, these should have been on, this is three different, I should have put them on three different lines here. So why the Southern Sea Otter Recovery and Research Act divided the Republicans so strongly? I haven't looked into it. I'm sorry, I don't know. But you can, in terms of your data, whatever your data is, go back and analyze and see what it is that's really the main reasons for the clusters. And so these are the main reasons for those clusters. We can, again, if you've got really good eyesight, look at it over time to see how things are changing over time for each year. If we just focus on the years 1994, 97, 2000, and 2009, you can go ahead and see that in some years, we've got the Democrats are here. So, the blue, remember, is the Democrats, the red is the Republicans, and you can see there are things connecting the two groups. So in years past, there was actually clusters that included both Republicans and Democrats. So that has occurred in the past. What the arrow indicates is it's indicating overlappings between, I forget which one, whether it was G1 or G2, but the arrow is indicating in terms of people who are in one of these two groups. We'll circle this one, that means it was probably G2, that there were people that were in this. Now, they may have been split into other places as well, but people that were in this cluster were also in some of the overlapping clusters here who had been considered to be more moderate. So you can see the progression over time as well if that's one of the things involved in your data. So any questions about this second application? Let's take a look at our main interest, and that is, well, my main interest is biology, so application 1, and it was the main thing in the paper, is the breast cancer gene expression. So what here we do is microarray data, and so we'll take a look more at what microarray data is soon. We will compare two data sets, so we'll be able to see that the pictures from two very different data sets actually do look fairly similar. Again, we're going to use Pearson correlation as our distance. Our filters will be this L infinity centrality, which is given a point in your data, then you'll take a look at the maximum distance. So if I have a lot of data here, I will take the maximum distance between. So for this X right here, the maximum distance would be that. So it captures the structure of points that are far removed from the central or norm. And, cancer is kind of, your tumor cells are kind of far away from the norm in terms of things that they are measuring. So there were two filters. One was this L infinity centrality. The other one was for the NKI data. We have survival versus death. So that's either the patient survived or they died. So there will be no overlap between these things here. And then for the other one, it's whether they relapsed or whether they didn't relapse. So usually these data sets are over a period of 5, 10, maybe 20 years, depending upon your data. And so you might just know that they have relapsed or not, or you may know whether they've died or not. And so that's the sort of second. So the two filters are these. Here, we will definitely get some overlap between our data, but here we will separate at our data because you either survived or you didn't. Nobody did both, at least during the time of the study. And then the clustering will again be this single linkage clustering. So let's take a look at microarrays. So I'm going to give a very basic stuff. I will add additional links. I forgot to add some links. There is some nice places where you can learn more about microarrays. So I will add that to the additional reading section for this week's stuff. But basically, microarray uses the fact that if you have DNA, single-stranded DNA. So let's start off with some single-stranded DNA. That means I might have a T here, an A, a C, a G, maybe a T over there. And so these things, they like to bind to the complementary strand. So we could have another strand over here with the complement. So if this was T, A, C, G, T, then the complementary, this one would want to bind to an A. T normally binds with A normally binds with T, C normally binds with G, G normally binds with C, and then we'd have this one to bind with A. And so we could then get these things forming base pairs, joining them, and so that's how DNA is happiest, is when it's base paired. So if you want to identify a sequence, what you can do is make it single-stranded. And then if you have a microarray, so we've got this microarray where each of these wells here will have, so in your well, you'll have many copies of the complementary strand. So we'll have many copies of the complementary strand, the single part right here. And then if this is complementary to it, when you add it, it will come here and bind. And so if you can notice that binding, if you can identify that it's bound, you now know that this sequence here is identical to that sequence. Oh, did I just say identical? Well, I may say identical, but I don't really mean it. In biology, when I say something is true, like A always pairs with T, no, in biology, nothing is ever always true. A usually pairs with T, but there are exceptions. And so maybe you have complete complementarity. So maybe maybe you've got a perfect match, but you might have a case where maybe this G is actually a C, or maybe it's a T, and you still do get some binding. So there is always, biology, and that's actually a good thing about biology. This is one thing that a colleague of mine has mentioned, is that when it comes to trying to cure cancer, you don't need to cure it 100%, because if you cure it 80%, your body will take care of the remaining 20%. So if you can just reduce things by 1%, that's sufficient. You don't have to do this 100% thing. So this, 100% matching can actually be a good thing in some cases. So there's a lot of leeway that goes on here. Well, in microarray data, you have to be able to notice when things bind. So what you have is you have these fluorescent dyes. So you might have a green dye or you might have a red dye. I just turned my green dye red, didn't I? Okay, so we'll turn keep our red dyes red and our green dyes green. But you can basically, what you do is you have your microarray right here with all these holes. And remember, all these holes already have DNA in it. They already have single-stranded DNA with different sequences in the different holes. You can then add the patient DNA, and if it has the patient DNA, say has the green dye, after it has bound, you can then wash out all the excess that has not bound, and then you will see a bunch of green things where it has bound, and you can then visualize it on the computer. But you'll notice that there isn't just patient DNA here, there is also a control DNA. If I have the sequence like, A, just a bunch of A's, versus if I have the sequence CG, CG, GG, CC, they have different binding affinities. So the chance of one of these sequences finding its pair down here, even if the pair exists, they have to find each other. So the chance of it actually finding itself will vary depending upon the sequence. It will also vary when you create this array. They don't have exactly the same copy number. So there are many copies of, so in a single well, there are many copies of, this single-stranded DNA sequence. So there's many copies, but you're not going to have the exact copy number, and they could vary by, I'm going to make up an amount, maybe 20%. That's probably pretty, they can vary dramatically between the different wells. That means if you want to compare two different microarrays, i.e., if you want to compare two different patients, if you're trying to determine what are the properties that will group a patient A as to one where you should give them drug A versus patient 2, you might want to group them into giving drug 2. You need to be able to compare their microarrays. So to be able to more easily compare microarrays, what you do is you put in competitor DNA. So you're going to have a control DNA right here. And so now the two DNAs will fight. And so you will have these two, both the red and the control labeled red, the patient labeled green. They will all be added to the microarrays. And so if for this particular well, if more of the control data matches than the patient data, then you would expect this thing to be more red because the control data is red, has the red dye associated. So if we have more of the control DNA, this thing should light up more red. And so when you see the picture over here, you can kind of see something, this sort of thing looks more red, so that means that there was more of the control DNA in it. Versus if there's more of the patient DNA, so maybe the patient DNA has more of a particular gene, and so if there's more of that, then you'd expect the thing to be more green. I think I circled the wrong ones in the preceding example. So if we've got more patient, we are here. If we've got more control, we are here. And if they are equal, then it would be here. And the coloring would be different. If it's more patient DNA, it's going to look greener. If there's more control DNA, it will be redder. And if it's about equal, it'll be, a mixture between the two. And so thus, if you have more patient, then that means that the patient had more of that gene. If there's red, then it would be downregulated. And I've given a simplified thing. We're actually looking at gene expression, which means we're actually looking at the amount of RNA that's been created. RNA also likes to bind. Or you can turn the RNA into copy DNA and use that. So there's a variety of different ways of doing the microarray. But basically, in terms of the gene regulation, you have a variety of genes that make RNA. So you've got your DNA that then makes RNA, that then makes your protein. And the different expression levels, how much RNA you have does indicate there is a correlation to how much protein, but it's actually not one-to-one. So how much RNA you make may not indicate, it doesn't indicate exactly how much protein, but there's definitely a strong correlation because the protein is transcribed from the RNA, but there's a lot of steps that go on here. But here we're going to be taking a look at the gene expression in terms of how much RNA was produced. If your gene is upregulated, so in this case you've got upregulated, so often you get more inflammation when you're being treated by cancer, your body is reacting to the drugs as well. So, and it's reacting to the cancer too. So you might get more inflammation, and so the genes related to inflammation are often upregulated in your patients, and so you'll get, the green stuff here. You might also get genes that are downregulated. The genes that, should have taken care of the cancer might be downregulated, and so then you'd have more patient DNA. All this, however, is done basically statistically, stochastically. We have, when you bind on, I said there's a competition, and so I said when you have more DNA, it will look green. When you have more patient DNA, it should look green. Well, that's statistically distributed. You might have more patient DNA, but maybe for some reason the control DNA got there first, and so this is all noisy data. There's a lot of noise in here. Just because it's green doesn't mean it should have been green. There could have been noise that pushed it up to being green. Maybe it should have been more red. Maybe the red should have been more green. So lots of noise. Data is often noisy. Any questions to my simplified description of microarray data? Okay. So if we take a look at our microarray data, this is basically what it looks like. If I go across a single line, so if we take a look at this white line right here, that is a single patient. So this would be patient 177 maybe. I don't actually remember the exact patient number, but it would be that patient number that we can take a look across. And in this particular paper here, they tried to find the 70 most important genes that distinguished the patient DNA apart from the control DNA. Although when I say the control DNA, their control actually was, so there's a lot of different, when we go and use the control DNA, there's different types of control DNA. You might just be looking at the frozen samples. So in one of the 2005 papers, they went back to 1995, tumors that were frozen, and then they extracted the DNA from that, and that was their data. And then they didn't say where their control came from that I saw. I never trust a mathematician saying it wasn't there, maybe I missed it, but quite often you just buy your control DNA. So you might just buy control DNA and use that to intermix. In the case of this paper here, what they did is they took all their tumor cell from all the different patients. They took all the patients, mixed it together, and that became the control. So when you were basically having patient 1 competing against the average of all the different patients. And then the second line, your second data point would be patient 2 competing against all the patients. So the different controls will vary depending upon who's doing the experiment. So often you just buy it. Sometimes you have control patients, and that might be the average of that. It will vary quite a bit. So thus we basically have, so each line here is a data point. And our data point is going to be values. So each column here is the gene expression. So over here, we've got the patients. These are the rows. And on here, the columns are the genes. The genes, that is a column. So if I look down a column, I can see whether it is upregulated, so more patient, or if it's downregulated, depending upon the patient. And so we thus get a vector for each patient, depending upon whether the gene was upregulated or downregulated. And you can see that it's color-coded from minus 0.5 to 0.5. And then we also have the additional data. How long was the patient followed? So you can see that almost everybody was followed for at least 5 years. And you can see whether the cancer metastasized or whether the patient was still doing fine. So if it's a blue dot, the patient was still doing fine at their follow-up time, their total follow-up time. if it was red, then their tumor had actually metastasized. So that's the second piece of information that we have. Any questions about the data? Good, because we are now going over time, but let me just show the results. So these are the things, but let's just continue just so you see the results. These are the results that we have here using the two different things, where we've got those that survived versus those that did not survive. And notice we've got the two different data sets. What survived, we have something that looks very similar in the two different data sets. And they are very different data sets, but you can see some similarities between the two. These are the results. They then said, well, what explains the results? So they then looked at coloring using chemokine activity to say that there was a correlation there. And so this is the same thing as on the left, just colored differently. We will now look at software in the next class on Friday, and I may say a few more words about this data set as well. So see you Friday, same time.